Good day to everyone joining us and welcome to today's Xtalks webinar. Today's talk is entitled Creating Digital Biomarkers with Real-World and Existing Data. My name is Sydney Perlmutter and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers throughout the presentation. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to them throughout the presentation. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank VeriSense by Shimmer Research, who developed the content for this presentation. VeriSense Health was launched earlier this week as a spin-off from Shimmer Research, the wearable technology services and sensor manufacturing company, to focus on digital health. VeriSense Health is a digital health software and data management company. It does not produce its own sensors and will work with any qualified digital health company. VeriSense recently launched the Digital Health Panel, which is a longitudinal study capturing device-agnostic, ground truth digital data and combining it with real-world and patient-reported outcomes data. Leveraging its Digital Health Panel, VeriSense Health provides a wide variety of services to meet sponsors' digital health data collection needs. And now, without further ado, I'd like to hand the mic over to our moderator, Geoff Gill, who will introduce himself, and our speakers will introduce themselves afterward. So Geoff, you may begin when ready.
Hi, my name's Geoff Gill. I believe my webcam should be coming up. There it is, shortly. As Sydney mentioned, I'm moderating the panel. As some of you may know, I've been actually working to accelerate the adoption of digital health technologies for quite some time. I did this because digital health, I believe, has the potential to transform medicine from reactive treatment of acute illness to proactive management of disease, where we actually prevent people from getting acute. From my perspective, the biggest challenge in digital health is the adoption and acceptance of digital biomarkers, saying what people, whether someone's getting better or worse, and whether you need to intervene or not. I co-founded the Open Wearables Initiative some time ago to encourage the development of standard biomarkers through open source algorithms. But I think maybe the biggest challenge is that we're really missing the data needed to develop and validate these biomarkers. I think we can address this gap by reusing a lot of the data that we're actually generating today and also connecting it with real-world outcomes. And that's why the topic of this webinar came about. I'm currently transitioning from my role as president of Shimmer Americas to become the full-time CEO of VeriSense Health, where we're trying to fill the gap with the VeriSense Digital Health Panel. Today's discussion, as I mentioned, is about the reuse of data and real-world evidence. We've got a great panel, trying to keep it interactive. Knowing my fellow panelists, I don't expect this to be a huge challenge, but now I'll let them introduce themselves. Dan, perhaps you can start it off.
Yeah, hey, thanks so much, Geoff. And I don't know how often it is that we get to participate in events by a company that was spun out less than a week ago. So way to hit the ground running, I suppose. It sounded funny hearing it said that way. And congratulations on the progress, both with Shimmer and now with VeriSense. So I'm Dan Karlin. I'm a psychiatrist by training. I'm also boarded in addictions and clinical informatics. I have a faculty appointment at Tufts University in the School of Medicine and a medical staff appointment at Montefiore here in New York. I'm currently the chief medical officer of a brain health focused pharmaceutical company called Mind Medicine, or MindMed, which also has a digital division developing companion products to alongside, sort of live alongside our drugs and to potentially be used in clinical trials for the drugs. Those products all have sort of an FDA facing component to them, so I'm interested in regulated digital products. Prior to that, I ran a company called Health Mode, which was entirely focused on digital measurement for clinical trials and clinical care. I also developed a product called Nightwear, which is a treatment for PTSD nightmares that exists entirely on an Apple phone and Apple Watch. And it's a closed loop digital therapeutic that now has FDA clearance and is a marketed product. I spent some time at Pfizer where I developed psychiatry drugs and built out what we call digital medicine at Pfizer and the Pfizer Innovation Research Lab. So, you know, when I hear, you know, Jeff talk and think about the topic that we're addressing today, both the development of digital biomarkers and the use of real world data, there are really three core tensions that I think have to be addressed in order to progress the conversation and to progress the development of these things. And the tensions, you know, three of them, and we could come up with many different ways to structure the question or the problem. But to my mind, the three main tensions are reality versus models of reality, right? So reality is impossibly complex in many dimensions. And so we're constantly, both intrinsically in our brains, but in our way of describing the world, building models that are comprehensible, that give us tools in order to try to interact with reality in a meaningful way. So effectively, all of our understanding of human biology depends on these models that necessarily dimensionally reduce a complex reality into a more comprehensible system of understanding. Okay, that's one tension. There's a tension that has existed, sort of been recognized in informatics as long as we've had a specialty called informatics, which is readability versus reusability when it comes to data. So, you know, in my field, psychiatry, arguably all of the novels in human history were written to try to understand psychology and psychiatry, right? You can get an incredibly deep and rich understanding of people through reading all of the novels of human history, if you have the time. Not super useful for designing a diagnostic methodology. So on the very opposite end of the readability reusability spectrum, you get something like the DSM, right? Highly reusable, but not very rich. And so in all of our data, we have to consider richness versus reusability. And when we aim too far toward reusability, we almost necessarily sacrifice richness, which is of course what makes it a tension. And then the third tension that I think is important, and I'll try to tie these together because they may seem a little disparate, is when it comes to validation of a clinical measure, we always have to consider valid against what standard. And the two ends of that spectrum, for the purpose of today's talk anyway, we'll call concurrent criterion validation, which is to say some new way of measuring matches up pretty well with an old way of measuring. And it's of course the quickest way to get to validation is concurrent criterion, if you're going to bother at all with validation, is concurrent criterion validation, because you have something that's been accepted as a meaningful measure of disease, and now you've got something new that comes to the same conclusion in the same person at the same time. It's pretty cool, right? But there's a much more important type of validation that takes a lot longer to get to, and that's predictive clinical validation, right? And so predictive clinical validation means there is a known and established clinical state that ought to be itself measurable, and this new way of measuring the person gives us data about the likelihood of the occurrence of the known clinical state. So it's working backward in time. Let me try to tie these together with an existing digital biomarker, and I hesitate to say validated, and I'll tell you why in a minute. So we've known about the idea of blood pressure for a really long time, right? Probably Hippocrates had some sense of blood pressure, right? We can detect blood pressure when we check a pulse. If it's very high or very low, you can tell just by feeling someone's radial pulse. We've known for a long time that a very, very low blood pressure was bad and was highly predictive of a bad clinical state, like loss of consciousness or death. And we've known for less long, but for longer amount of time, that very, very high blood pressure, also bad. So 300 systolic, right? You start to have immediate clinical events from that. Through the repeat measurement of blood pressure using sphygmomanometer and auscultation, we got better and better at detecting blood pressures in a range that wasn't extremely low and wasn't extremely high. And for the most part, these were analog measurement devices where whoever was taking the blood pressure would watch an analog meter, and in their head, based on what they were hearing when they heard sound changes, would record a number, right? That's all analog to digital conversion is. And so we used human AD conversion to get to a reportable number, a reusable number, right? And by measuring blood pressure over and over and over again in a lot of people and watching what happened to them clinically, we came to develop a clinical state called hypertension, or essential hypertension, which just means we don't know what causes it. And that definition of a new clinical state gave us something to treat, where we discovered that if we treat the new clinical state, reduce the blood pressure, treat someone's hypertension, that their clinical outcomes then change. They are less likely to have the known clinical outcomes, renal damage, retinal damage, et cetera, heart attack, stroke. And so literally through repeat measurement, converted to digital, data reused, we devised a clinical state that can be treated to that reduces morbidity and mortality. This is the goal of digital biomarkers, right? It has to be the goal of digital biomarkers. It has to be about looking more closely, a lot of times and a lot of people, to try to identify novel clinical states. And those can either be subdivisions of existing known clinical states, or it can be truly novel, usually earlier in time, temporarily earlier clinical states. Now, what's the problem there? Why did I hesitate to say validated? And this is, so there we saw readability versus reusability. We saw a model of the world versus a reality, very useful model. And then as we progress forward in time, what we've discovered now is that because we have the ability to do at-home blood pressure monitoring much more easily, we can now sort of know reliably that in-office sphygmomanometry is not as valid as we thought it was. It's not as reliable a measure of people's at-home blood pressure as we expected it would be. So now we have to kind of rethink this and say, well, we've probably been both over and underdiagnosing actual hypertension, because the way we were measuring it episodically versus continuously had a measurement error, had a measurement bias. So there's a model that wasn't what we thought it was as it related to reality. We thought in-office sphygmomanometry was a good model to understand real-world walking around blood pressure, and it turns out it's not as good as we thought it was. So as we sort of think through the various tensions here and try to come to terms with what that demands of us as people building measures, I think that we have this amazing opportunity to try to more reliably create more reusable data across measurement paradigms. And so, what do we do to do this? Well, we have to try to figure out how we can all measure the same thing in the same way across a lot of people and share those data between us, right? And so that means things like when we're designing studies to validate digital measurement, making sure we're going as close to the sensor as possible, right? So rather than relying on what could be proprietary algorithms to pre-process data and only collecting and reporting on those process data that may vary from manufacturer to manufacturer, sensor to sensor, phone to phone, app to app, whatever else, the more that we're able to capture and report data that are very close to the sensor, not as people say raw data, there's really no such thing as that. Everything's got some degree of processing, but as little processing as possible before it's collected. And you can do whatever processing you want downstream. Of course, we all do that, right? That's how we devise algorithms to try to make meaning out of the measurement. But if we're able to share and report close to the sensor data that's well annotated so that we understand what was going on when those data were collected, who it was collected from, what the sensor was, where it was worn, what the person was asked to do, then we're better able to downstream reintegrate data and start to get to something like what I was describing with the evolution of the definition of essential hypertension, but for other conditions that are fundamentally phenomenological. Because in the end, the absolutely vast majority of what we're measuring when we talk about digital is the behavioral outflow of an illness, of a person, right? We're really talking about what we would call phenomenology. And so in the end, digital biomarkers are about looking at fine-grained phenomenology, being willing to accept that by looking very closely, we might actually break existing models of disease, might break existing models of how people are, how they work, and being willing to accept that those models might need to be broken in order to get to better measurement, better disease definition. We need to do that with an eye toward prospective clinical validation, right? Predictive clinical validation. And we almost certainly need to do this in largely pre-competitive environments if we're going to get to meaningful measures of people. And I think I'll probably wrap my intro, I guess, there. But anyway,
that was great, and Dan, you've raised a huge number of topics. I think that I may turn it over now to James, and who is actually in the process of doing a number of studies where he's looking at reusing some data that were captured from an original study and now reusing it. And maybe you can talk a little, first of all, give a little bit of your background, and then talk a little bit about your experience there and what's important, to you on a very practical level.
Absolutely. Hi, everybody. My name's Dr. James Connolly, and as I'm sure you can tell from my accent, I'm from Ireland. I'm a lecturer in computing, and it's within artificial intelligence, big data, and of course, connected healthcare. And I supervise various Ph.D. students working through projects using wearable technology to improve the endpoints and the outcomes that come from data. And as Dan has already said, data is really important to us. We're getting to a stage where we're trying to get as close to the data as possible, and we've been typically trying to do that over the last number of years. And we've come to a stage where we find that some of the sensors that we use and some of the information that's coming from them just isn't as accurate as we expected it to be. And we're starting to look at different measurements and different types of systems that we can use. We were looking at Euclidean norm -1, which is a typical type of measurement that uses the GGIR package, but we also would use the 6-minute walk test, and we're beginning to look at what's called the maximum 6 minutes activity as well, which is a new measurement that we're finding seems to be able to capture movement a lot better. It measures the maximum amount of movement that happens within a time period. So it could be within an hour, it could be a day, it could be across a week, but it's a better way of detecting light and sedentary movement. And we'll probably talk about that more now whenever we're within this webinar. So Jeff, I'll leave that until we get some nice questions.
Okay, great. And I would say that one of the things that James is doing is he's using this algorithm called GGIR, which is an open source algorithm, which, hopefully we'll get to talk a little bit about that and the advantages of that, in that everyone can use the same exact algorithm to process data that can be independently measured, and no matter what sensor or device you're using. But I'll leave that as a teaser, and then move to Bill. And Bill, maybe you can talk a little bit about sort of what's happening in the sort of regulatory big picture environment in this, both with sort of reusing data and with real world data.
Sure, thanks, Jeff. Hello, everybody. I'm Bill Crown. I'm a distinguished research scientist at Brandeis University, and before that, I was Chief Scientific Officer of Optum Labs, and before that, I was in the consulting world, in healthcare consulting and life sciences. Ran outcomes groups for a couple different companies, really all focused on analysis of real-world data to generate evidence around comparative effectiveness of different treatments. In my role at Optum Labs, I had the pleasure of being able to put together a really massive real-world database of about 130 million covered lives of claims data, with 85 million lives of electronic medical record data, and then linkage to mortality and sociodemographics, and county level data from the Area Health Resource File from AHRQ in the US government. And that data set really sort of became a rallying point for academics around the country to analyze the data to generate evidence. And I'm a real believer that the wealth of data that exists in real world data sets has the opportunity to generate evidence much more quickly than randomized trials. That isn't to say that randomized trials aren't useful, but there's a great ability, I think, with observational data to generate really credible and useful information when appropriate methods are used to analyze those kinds of data. There's a lot of interest in this area now from regulators, and it stems in particular from the passage of the 21st Century Cures Act, which requires the FDA to develop a framework for the incorporation of real-world evidence around new indications for previously approved treatments, as well as safety surveillance. And the FDA has been in the safety surveillance business for a long time, particularly with the Sentinel network, which is a massive national claims database that they use to query the safety of existing products on the market. But this idea of looking at new indications for previously approved products and using real-world data for making regulatory decisions is a new thing. And that's really caused the FDA to shift its mindset from really being solely one of randomized trials to two randomized, well-controlled studies, as they put it, in improving new products to thinking about the role that real-world data can offer there. And that's made them think about whether it's possible to generate causal inference like they would have with a randomized trial from observational data. So they funded a very large national study out of Harvard called RCT Duplicate, which is in the process. They've pretty much finished emulating the 37 different randomized trials. 30 of those have been completed already, and this study is using claims data to emulate the inclusion-exclusion criteria and endpoints and follow-up periods and so forth of those trials. And then there's 7 trials where the products are already on the market, but there's a randomized trial going on to test the real-world clinical effectiveness of those products. And the database studies are being run and are being run more quickly than the trials, of course, and predicting what the trials are going to say when the trials are actually finished. It's been really kind of remarkable to look at the evidence and the degree of agreement between these observational studies and these randomized trials. But the one place where I think there's a huge gap is in the real-world evidence space is digital therapies and evidence from digital devices, where it often isn't possible to identify those devices in claims data, particularly when they're new to the market. The ability to be able to bring that data in and link it we have that technology now with salting and hashing methods to be able to bring data in and link it up with existing data sets. So I find myself in great agreement, of course, with everything everybody has said, but particularly what Jeff started out with, where the idea of being able to reuse data, to advance medicine is I think very, very real and very exciting. Back to you, Jeff.
Sure. Yeah, and I was just going to actually sort of jump off on that. I mean, Dan made a comment about the need to have data that's close to the sensor, and maybe James, you're actually trying to do this. I mean, if you don't get sort of raw data or data that's very, very close to the sensor, are you able to really reuse it? In other words, if you are dealing with something like steps or whatever, is that something that can be effectively used?
Yeah, good question, Jeff. And as Dan says, we can never get this stage where it's totally raw. There's always going to be some type of filtering that's been applied to the data, whether or not it's something that goes on at the firmware level or maybe it's something that the sensor does before it's transmitted to the host computer. But essentially, the key goal of what we would really like within any type of measurement is that the data doesn't have a filter applied to it, because the filtering really reduces the granularity of the data. Sometimes it creates summary information of it, or sometimes it maps out outliers, or sometimes it tries to improve it unintentionally to make our lives a bit easier. But in doing that, it removes the usefulness of it, so that essentially it means that sensors become almost like they're standalone. We can't really compare one sensor against another because we don't know what type of algorithms have been applied to them. And that means that future data comparisons then become, basically we can't do that anymore, because the filtering just makes them more useless to us. And the other thing is, if we wanted to then apply our own filtering technology, say for example, if we wanted to create an AI system, then filters also make that a bit useless for us. So the important thing for us then is to get as close to the raw information as possible. And what we mean by that in terms of technology is that we don't apply anything to the sensor data, and it's transmitted as cleanly as possible, I would say. So it means that, you know, whether we use say an Apple Watch, or if we use a VariSense sensor, or if we use any type of IMU sensor, that the data should generally be representative of the raw sensor itself and not of the algorithms that were used to process it.
No, I know personally, people often, you know, object when I say raw data, and I always come back to the definition that I have, which is, to my mind, raw data is something that can be independently physically verified. And so if you have something like acceleration and you can measure that, you can independently verify that. And so as long as you're starting there, that to my mind is kind of good enough for raw data. But at any rate, you know, I think, you know, Dan, maybe kind of coming back to you. I mean, I know there's been a lot of talk about sort of, you mentioned pre-competitive cooperation is really important. You know, how much of that is actually happening?
Less than we'd like to see, I think, is the easiest answer. There are some examples of projects that are more collaborative, and you know, going back to there's a project called Watch PD that Ray Dorsey was at the middle of, but where I know that at least Biogen and Pfizer were involved. I believe another pharma was also involved in that. So, that was a truly pre-competitive effort. Transcelerate has a digital biomarkers effort that brings pharmas together. I haven't been involved in that in a little while. My little current biotech is not a Transcelerate member just yet. CTTI has been involved in trying to establish some pre-competitive things. I don't know that those are directly data collection projects. I think those look a lot more like you know, policy papers and best practices for research and things like that. Though, of course, anything that gets folks aligned in the research world is likely to lead to the ability to have better data integration and better pre-competitive efforts, even if they're post hoc post pre-competitive efforts, where maybe the research wasn't conducted jointly, but there are data that are interoperable and integratable in certain ways. And then put a plug in for the Digital Medicine Society. I'm the chair of the board there, and we formed, I don't know, now 3 years ago. You know, under Jen Kolczak's leadership, the Digital Medicine Society had absolutely outsized impact for its age. But I know that there are some projects under the auspices of DIME, like a collaborative itch and scratch detection project using wrist-worn accelerometry to look at nighttime scratching behavior that's been going on, that has had a number of participating organizations. DIME, beyond direct sort of prospective research, is also working at establishing best practices across a number of domains in digital medicine. So there are some more established efforts, and there certainly are growing nascent efforts to try to do both prospective collaboration and to try to get data to be able to be integrated after the fact. And I think this speaks to what everybody's saying, which is that if you have pretty close to the sensor, pretty well annotated data, and you have some ability to integrate across those metadata, the annotations, then you start to get to some really powerful ability to expand into larger and larger data sets. And at some level, particularly if we're going to be applying ML and AI to these data, there is something to be said for data that were not collected in the same environment with the same protocol, because the AI, while artificially intelligent, is not intrinsically intelligent, and it's very prone to learn on things we didn't realize we were teaching it, that are just artifacts of how a study was conducted, how a protocol was executed, things happening at a site. I mean, it's one of the reasons that AI has struggled to find clinical meaningfulness in medicine, because so often these algorithms are being trained like in one system and are learning to, rather than learning to read the X-ray, they're learning to read the metadata that's embedded in the X-ray. And so, particularly when applying AI, it's going to be increasingly important to be able to integrate data sets from across a number of different data collection sort of environments.
Mm-hmm.
I'll just jump in quickly. There's an interesting variant on this, which is for a long time, life sciences companies have been using doing secondary analyses of claims data for primarily for to support commercialization of their products. So they do it also for sort of understanding burden of illness and so forth. But it's interesting because it's sort of in a way, it's almost like a pre-competitive thing, but they do it for competitive reasons. So there are major vendors like Truven and Optum that license these data sets, and the big pharma have warehouses of these different databases that they use, and they analyze them to look, compare the comparative effectiveness of their products and support their value propositions around pricing and so forth. So I think it's there are examples certainly where existing data sets are used by across the industry, but to support their support the commercialization of their own products.
And it's, we tried so hard for a bunch of years to try to make medically meaningful discoveries from claims and EHR data, and it's really, really hard. I think partly because of it's great for marketing for sure, and for targeted marketing, and even for things like indication selection. I think one of the struggles with that sort of data, if you're trying to actually discover new things about the world, is that those data are so locked into the existing ontological structures that there's, it speaks to the predictive versus concurrent validation problem, which is you're never going to be able to, well, I won't say never, there's some, I'll offer some hope. It's very hard to say, look at subtypes of major depressive disorder in any meaningful way beyond the existing nosologically defined ones, using systems that are built to express things in terms of the existing nosology. So that's one of the weaknesses. Now, that said, as we get better at both processing free text and making reusable computer meaningful reusable data out of free text versus just readable readable data, I think we may actually be able to use these sorts of integrated EHR data sets, de-identified as they are, and all the other weaknesses in them, to maybe start to be able to at least posit places to look to go make meaningful medical discoveries.
Yeah. And I do think there's the opportunity, even though those data sets are made available commercially in de-identified form, there's upstream identifiable data. And so it's possible to bring in, things like sensor data and link it to upstream data in those kinds of systems, and then de-identify it and make it available for research purposes.
Yeah. I guess what I would say, I wanted to kind of bring it back to, because we wanted to talk about digital biomarkers in particular. Obviously, there's a huge amount of work going on in real world evidence and building that whole infrastructure. But Bill, you mentioned that the sort of the gap there seemed to be in digital data, digital health data. Do you have a sense of why that's the case?
Yeah, it has to do with for digital data that's being sourced by FDA approved products like glucose monitors and things like that. There's a lag in the time from when that approval happens to when there's a approved code from CMS that ends up showing up in these databases. And so without the code, you can't identify what the device was that was used to treat the patient. And then eventually, over a period of time, the coding appears in the database as CMS issues the device codes. But I think that's really been the major limitation for the analysis of device data by academic researchers are the coding issues.
But I guess I'll ask the question. I mean, we've been talking about how you need the raw sensor data, or whatever you want to call that data, and we don't yet have digital biomarkers, so it can't really be used as a medical device, which means it's not going to get a CMS code, which means you can't share that data. And what do we do about that?
Yeah, I think well, there's sort of two sides to that. One is when it comes to this issue of having the data sort of as close to the source in its original form as possible. The FDA is all over that, and it's been a real pushback that they've had with all these kind of commercially available data sets, that they want to be able to understand the source and the provenance of the data and to it before it's included in these databases. So I think sort of the sentiment of the panel around having the data in its original form as possible is very in line with what the regulators are looking for. I think in many cases the devices aren't necessarily things that we where there's a regulatory approval being sought for the use of that device in a treatment, but rather they are measures of sort of patient performance that we would like to be able to link up with other data that we have in order to put it into context and to be able to measure outcomes and so forth. And there, I don't think there's any issue at all being able to bring that data in, because it's just a matter of linking it at the person level with existing data sets. There's a challenge that researchers always have with these data sets around HIPAA regulations that are designed to minimize re-identification risk of patients. And the more granular the data gets and the more rare the condition the patient has and so forth, the more things you have to sort of give up in order to mask that patient. And that's a challenge. And I think we need to work on that, doing with methods like differential privacy systems and so forth, where we can maintain the granularity of the data without sort of losing that information. But I'm not sure that it's necessarily a huge issue in terms of being able to bring in digital markers and link them up with other data sets. It's only when it's more of an issue when there's a regulatory approval attached specifically to the device, and then there's this lag time because we have to wait for the codes to show up.
Yeah. So I mean, it's to my mind, it's kind of where it gets incorporated in the data stream. It's like you're not getting the digital data through the sort of the normal EHRs or whatever. That just doesn't really exist, but you can link. The idea would be to link them, and I think that's where we are, is trying to get this linked. I mean, I'll go back to James again, who's trying to work with some specific measures. He's got some data, both some clinical observation data and digital data, which is outside the sort of the healthcare system, and that's where he's working. I mean, and to my mind, that's through Shimmer and also now VeriSense, we're seeing, I mean, that's where a lot of the work is going on right now. It's kind of outside the clinical stream. So it feels like we need to somehow figure out how to marry up the digital data with these clinical streams that have huge amounts of really rich and arguably objective and and accurate outcome data. But I mean, James, kind of working in the trenches, are there ways that you can access any of these data, or talk a little bit more about the sort of the process that is going on today?
Yeah, that's a very important question, because as a researcher, as you say, working in the trenches, whenever we go to use any type of hardware at all, of course, we've got the problem of validation, where we've got to assess the accuracy and reliability of what it is that we're going to do. And really, what we need is some type of maybe like a repository where we could globally have this large place where we can go and we can get the raw information, and it can save us a lot of time and effort to have to validate this against accuracy and reliability. If you think about before we even get to the stage of using our data at all, I mean, it takes months to apply for ethics and then to test the systems against state of the art, and then to validate the data, and then to measure what's an acceptable level of error before we even begin our tests. So really, this if we could bypass that, that would save months of work. And why can't we do that? Why can we not use an IMU sensor repository that's been used on, say, COPD patients, or perhaps if we've got hundreds of stroke patients who've been tested for rehab, then why can we not use that instead? Why do we have to do it ourselves? There's no point in reinventing the wheel. So as a researcher, I think that is something that we really need, and it would be a definite game changer in terms of research. That's my opinion.
Great. Dan, do you have any thoughts on this, or? I think you're on mute.
Yep, there we go. First one of the and I unmuted only to say no. I think that was really well covered and thoughtfully.
I just kind of add in. I think this was prompted by Dan's mention earlier about machine learning, and I was thinking of and the operation on the text data and what benefits that might bring. And we did a study in Optum Labs. It was about a 4-year project where we started out with just sort of trying to predict early onset of dementia, with the idea that we could intervene with patients and try to keep them out of long-term care settings and help them at least plan for the financial consequences of that and so forth. So we started out with claims data, and we had a group, one of the academic collaborators, that had a literature-based model that they produced. And the machine learning, just operating on the claims, found all of the indicators in the literature and then a whole bunch of other things. But it didn't do a whole lot better in terms of predicting than a simple logistic regression model did that the academic group was using. And then we linked in electronic medical record data, and we used more sophisticated methods. and the models just jumped in performance. It had to do with things like, as Dan was mentioning, the text, the raw text mentions of things like falling and forgetting things and so forth. And we created these word vectors that were very powerful predictors. And so I was thinking about the ability of sensor data, that measures things like gait, and having that in the electronic record, and then having machine learning kind of picking up on that. I have no doubt that that would be very, very powerful in a predicting analytics sense, and sort of predicting things like early onset of dementia. And it's just an example where the integration of these data and bringing in sensor data in particular there's not a lot of it there now, but there's certainly the potential to have a lot of it in a hurry, because a lot of it's being collected. It's just it's in all different places, and it hasn't been unified with other existing data sets.
Yeah, and I guess to Bill, what the problem is, that what we need is to have data that's been labeled. That's part of the problem, and the other thing is that we end up with AI systems that are overfitted into a particular type of data, and they don't have access to all of the other variations of data that maybe just isn't as as severe or of the patient's outcomes. So we need to have a more broader sense of the data so that we can train our systems better. Yeah,
I agree.
So I'm going to shift us a little bit and look at ask about how doctors and providers are actually going to end up having to use some of these things. Because I was reading an interesting article recently, actually just this morning, about the title was, nurses need to be at the forefront of digital health. And maybe we can talk about the doctors and the providers, how this is a whole new realm of things that they're going to need to deal with. And one of the barriers that I've heard is people don't want things showing up in the medical record that they don't know how to analyze, because 5 years down the road, someone might know how to analyze it, and suddenly, so well, you should have seen it there. But maybe Dan, I mean, as a doctor, you can talk a little bit about sort of how this needs to get integrated into the medical profession and into actual practice.
Yeah, doctors face the same sort of tensions that I was talking about before, right? There's time, just pure time. So a given visit pays a certain amount, and you can do only so much in the amount of time that visit requires. And so unless you're looking at metrics that are likely to change the care you provide, it's not a good use of time in the visit. And so, medical meaningfulness, it's the same thing with regulatory, really, right? Regulators are not going to stand in the way of us bringing forward something that we've proven to be medically meaningful. And you hear a lot about doctors being reluctant to change practice patterns and stuff like that. My experience is not that. My experience is that when confronted with something that meaningfully changes our ability to keep people healthy or get them healthy, I'm happy to adopt new things. But there's a pretty high standard of evidence to change the standard of care. And so we come back to that medical meaningfulness thing. But fundamentally, I think the deeper problem here is that as I said before, what we have the ability to do mostly with digital measurement is to look more closely at phenomenology. And in any field, and every field in medicine has these, in any field with phenomenological diagnoses, we fundamentally have models that are inaccurate, right? So in my field, calling depression a disease misses the fact that it's probably 1,000 rare diseases, maybe 10,000 rare diseases with more unified underlying biology. But because we've defined it based on a very coarse phenomenology, if we then go look more closely at 1,000 depressed people with digital measures, we're going to start to see that our model's broken, right? That the fine-grained phenomenology clearly clumps people into different subgroups that likely represent different underlying biology. How do you deal with that? right? Because we don't have medicines that are specific to those underlying subtypes or those groupings. So it really requires pushing further back away from the doctor into drug and device, treatment device development to say, how can we work all, as a specialty, as a field, as digital medicine, as individual medical specialties, to try to subtype phenomenological illnesses, to develop drugs targeting those subtypes, to be able to get regulatory approval for those subtypes, even though those subtypes may not be a meaningful diagnostic entity as of today. This requires all parts of the system to push forward in a unified front, because if I go to FDA and say, look, I've discovered this depression that 5 out of 100 people with MDD cluster this way, and now I have a medicine to treat it, they'll say, well, that you just defined a cluster to get your medicine approved, right? But if I can convince, say, the APA to include that kind of cluster in a diagnostic methodology, now we're working together to say, no, this is a meaningful diagnostic group. We can develop drugs to target it. The drugs are more likely to work better because we've got a selected group rather than a big heterogeneous group. And the reason in phenomenologically defined illnesses that we don't have very many curative treatments isn't because drug companies aren't trying to cure disease. It's because there are a bunch of different diseases that can't be cured by the same mechanism. So we end up just treating the phenomenological symptoms we're able to observe. Breaking mental models of the world that we rely on to get through our day is a really hard thing to do. And so that's the challenge for all of us, is being willing to say, yes, these models are useful in the current world. We have to treat to something. We have to develop drugs to something, but we have to be willing to and eager to, in many cases, to say, but what's a more useful, a more fine grain? We're saying closer to the sensor, closer to the biology disease definition, even as we know that it's still a phenomenological disease definition.
So on that note, I'm going to change it again and say, all right, we promised the audience here some kind of practical advice, and is there anything, if people want to believe in real world data, believe in reuse of data, what are some of the 2 or 3 things that that your advice to them would be? And I don't know if anyone wants to go first, or I mean, I can start it off. My number one thing is that if you ever want to reuse data, you need to have the raw data sort of as we've defined it. It's obviously not exactly raw, but it's as close to the sensor as you can get, and can be independently verified. I think that's one. And then I think you need to think about, identifying maybe a mechanism that allows you to share this data. I mean, one of the things that, I've worked with a number of teams in clinical trials, and the last thing at the end of a clinical trial, the last thing they want to do is go through a whole nother process to make their data available to someone else. Right? It's a pain in the neck to get that data there. So I think trying to figure out how to what the mechanism will be up front to do that, and to establish that up front is, I think those are the 2 sort of pieces of advice that I would have. And then I don't know, James, do you want to?
Yeah, I was just going to say, yeah, I agree with that. But the other thing that's really important is that we need to understand the background to the way the data was collected in the first place. So, for example, if we were going to do a 40-meter walk test, then we need to be sure that we define the parameters that we used to collect that data. Because if we don't all use the same protocols to collect it, then of course the data is going to be different, even though the test that we examined or that we collected the data from would be the same. So for example, the 40-meter walk test could be 1 times 40 meters, it could be 2 times 20, it could be 8 times 5 meters. So that's going to change the data and the shape of the data if it's not all the same. And then that means that the underlying patterns of the data is different. So that's really important, that we have a protocol that we use to define how we collect the data in the first place.
Bill, you've obviously reused huge amounts of data. So you want to talk about what enables that to be the case, and what are the, and also on the real world data side?
Yeah, it's I'm really optimistic because the 21st Century Cures Act has really changed the game in terms of the FDA interest in real world data, and that has spilled over. It's global now. Regulatory authorities around the world are interested in this, as are health technology assessment groups that make decisions around drug coverage and pricing and reimbursement decisions. So this is something that's here to stay, and there's a lot of existing data infrastructure that on sort of general patient populations here in the United States and elsewhere. But what we don't have is sort of the sensor and device data kind of brought together, in one place, where we can then take it and link it to those existing databases to be able to bring in sort of all the information around the context of the patient, their medical comorbidities, and other diseases that they have, and so forth. And from a technical standpoint, that's an easy problem. As long as we have identifiers, in the device data and identifiers and these other data sets, we can link it without having to exchange names and addresses and social security numbers. We can just hash the identifiers in both places and link the hashed IDs together, and then we have a de-identified data set that we can use that has the sensor and device data in it. So I think the first, a really big challenge is to centralize the device and sensor data so that we can then link it to other data sets.
Yeah, and having some method where we can control the connection between them all as well is really important. That's a real nightmare when it comes to trying to amalgamate data from several sensors together. What's really important is that we had some type of software system that could then do that hard work for us and ensure that all the amalgamation is happening accurately.
Dan, you have any closing comments?
Yeah, maybe a couple quick hits. Be proactive in consent in research. So get permission to share data, even at an identifiable level. People will often agree to it. So go as far as your IRB will let you, because I've not yet met a research participant who, when asked, can we share your data, says no. These are people who are participating in research for the sake of trying to help people with their disease or people like them, and almost always they're like, well, why you wouldn't share my data? That doesn't make sense. The whole point is to make these data useful. That's why I'm doing the effort. Pressure test your assumptions. So kind of be a baby, be a newborn when you start looking at any construct and say, well, what if this is just made up, and what if it is real? And then how can I dissect down further and get to the realest stuff I can? And I don't know, stop doing concurrent criterion validation. If I have to read another expensive digital way of getting something similar to the free and 30-second PHQ-9, like, come on, we can do better than that. So if you find yourself replacing something quick and free with something expensive, proprietary, and time-consuming, you're probably making a mistake.
Great. Well, I see actually we had a number of questions come in the last 15 minutes, and I have to admit that I was so involved in our discussion that I didn't notice them. But I will promise I will, send these out, and together we'll put together some responses to them, and I appreciate that. Again, apologies for not getting to it in real time, but we'll, I guess, Sydney, maybe you can take over and explain how people will actually get these answers. And thank you all thank my panelists for joining. I learned stuff, as I always do when talking to any of these people, and thank everyone for attending, and you know, we will see how this whole exciting area progresses. So Cindy, I will turn it back to you.
Thanks so much, Jeff. Good to see you.
Thank you, Jeff. And thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from Xtalks with access to the recorded archive for this event. Now, if we couldn't attend to your questions, the team at VeriSense by Shimmer Research may follow up with you, or if you have any further questions, you can direct them to the email address on your screen. Now, a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now I'm about to send a link in your chat box, and you'll be able to view the recording of this event on this page. And you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join us in thanking our speakers, Geoff Gill, Dr. Dan Karlin, Dr. James Connolly, and Dr. William Crown. We hope you found this webinar informative. Have a great day, everyone.
Thanks. Bye.
Bye.